Dear learners, in the previous module we learnt about the chemistry of conjugated linoleic acid and its natural and artificial food sources. In this module we shall learn about the bioavailability of conjugated linoleic acid and the effect of processing on its bioavailability. First we shall start with effect of processing on conjugated linoleic acid. Milk and milk products exhibit various structural, chemical and physical changes during processing and storage in their lipids. Milk lipids contain nutraceuticals like conjugated linoleic acid, sphingomyelin and butyric acid. Trans fats form in the lipids during the biohydrogenation in the rumen of cattle, but also be formed during cooking methods involving high temperatures such as baking or frying. As a study, it has been concluded that except for microwave heating, heat treatments and refrigerated storage of milk and dairy products did not cause significant changes in the trans fatty acid isomers and CLA content. To look into the metabolism and bioavailability of conjugated linoleic acid. Conjugated linoleic acid is actually a trans fatty acid, but as it is a natural trans fat which is found in various healthy food and it has health benefits also. There is a plethora of research which indicates that artificially created industrial trans fat has detrimental effects on the health, but naturally occurring trans fats possess different traits than the prior type and may benefit the health of consumers. Conjugated linoleic acid is simply an excretory metabolites or metabolic intermediates of rumen bacteria. It is normally produced in ruminants as for symbiotic purposes. Thus, it promotes health and longevity of the animals. It is also considered that CLA is produced by rumen bacteria as a factor specific for promotion and preservation of bacterial species in rumen. Mechanism of action. The possible action mechanism which shows that CLA can alter the body composition that involve metabolic changes which favors the reduction of the lipogenesis and the potentiation of lipolysis, accompanied by the oxidation of fatty acid in the skeletal muscle due to increased carnitine palmitoyl transferase 1 activity and action, or possibly because of adipocyte differentiation inhibition. Therefore, researchers have evaluated the action of CLA supplementation on the lipid and hormone profile and the activity of enzymes involved in the oxidation process. Studies have demonstrated that isomers 10 trans and 12 cis differently from the 9 cis and 11 trans of CLA increase lipolysis significantly in human adipocytes and also have the function of diminishing the synthesis of fatty acids. This would explain in part the possible action mechanism of CLA on the body composition. Although various studies were in vitro, the metabolic hypothesis to explain the body fat reducing action of CLA began based on control of the expression of gene involved in the differentiation of pre-adipocytes into mature adipocytes. In other words, the expression of these genes would result in reducing lipogenesis. It seems that the CLA interacts with the co-activator complex PPAR, increasing the gene transcription related to the differentiation of adipocytes, lipolysis through beta oxidation, mitochondrial biogenesis and insulin sensitivity, and collectively it is related to the weight loss effect. We shall now look into the physiological effect and health benefits of conjugated linoleic acid. Conjugated linoleic acid belongs to the family of omega fatty acids which are known to play vital role in various mechanisms for optimal health within the body. The conjugated form of linoleic acid is a collection of the different forms of naturally occurring linoleic acid. It may have health benefits in prevention or regulation of various conditions including cardiovascular disease, cancer, obesity, and may change body composition by reducing body fat percent. CLA is receiving considerable attention because of its supposed weight reducing, anti-atherosclerotic, anti-diabetic and hypotensive properties. Some researchers have also shown that CLA supplementation is linked with improvement in insulin resistance and influences osteoarthritis pathogenesis by modulating inflammatory factors. The fat lowering properties of CLA were originally discovered in rodents, and the debate continues to what extent they can be extrapolated to humans. Several mechanisms of action have been suggested, including a decrease in energy intake, an increase in energy expenditure, a reduction of adipocyte size, an inhibition of pre-adipocyte differentiation, an increased apoptosis of adipocytes, an inhibition of lipogenesis in the liver and adipose tissue, and an increase in fat oxidation in the liver and adipose tissue. Dear learners, to conclude this module, in this module we learnt about the effect of processing on the bioavailability of conjugated linoleic acids and its benefits on the human health. In the next module, we shall learn about the health benefits of conjugated linoleic acids in more elaboration.